Welcome back. So what we've learned so far is that you can breed alligators or pretty much anything, and you use that to figure out what the genotype of the parent is. But you really can't do that for humans, and yet we know how traits are inherited in humans, especially simple single gene traits. Whether they're dominant or recessive. And in fact, we can also determine whether they're sex-linked or autosomal. Remember from previous chapter, sex-linked, that means it's on the X or Y chromosome, really specifically the X chromosome, because the X chromosome has a lot of genes on it. Versus autosomal, that means on any of those other chromosomes, like 1 through 22, remember they have numbers. So we can use patterns of inheritance to figure out whether or not a trait is dominant or recessive and whether it's sex-linked or autosomal. So this is again a pedigree, and again I know you think about pedigrees as something that are something used for animals or show dogs or cats or what have you, but the same principles apply to humans. So let's take a look at this pedigree. This is a family tree. It's another way to say this is family tree. So any square is male and circles are females. Any kind of shaded in square or circle is an individual that has the trait that we're interested in. At this point, it doesn't really matter what the trait is, just that they have whatever trait it is that we're interested in. So we can look at this and say, okay, grandfather had the trait, but grandmother didn't. Again, it could be any trait. And then we have a few rules that we need to follow. So the rules are, if you're trying to figure out whether something is dominant or recessive, and it is the only thing we care about, if this trait carried by anybody that has the filled in, is that trait dominant or recessive, you have to look at certain patterns. And a pattern that really becomes obvious is that if the trait never skips a generation, then it cannot possibly be recessive. It has to be dominant. Dominant things, if you got it, you show it, right? So if it skips a generation, then it's recessive, or it could be recessive. So let's take a look at this and think about what I mean by skipping a generation. So with this pedigree here, we see that the grandfather had it, but the grandmother didn't. It's in this generation. It's not in this generation over here, but it is here. And that's the one that we're going to take a look at. So here, and then if we look at their children, so the way you look at their children is you follow the line. That's a child of that grandma and grandpa, that's a child. This one's not. This is a marriage into the family. And then this one. So it's in this generation and it's in this generation. The aunt has it and the father have it. Just because the uncle doesn't have it doesn't mean it skipped a generation. It's still in the generation, if you're following me. Once again, if we look on this side of the family, the grandfather nor the grandmother had it, none of the kids have it. So that's also not considered skipping a generation. What we're looking for with skipping a generation is where one generation has it and the next one doesn't and the next one does. That would be skipping. So, but in order to do this again, you have to really follow carefully the lines. So here we have this aunt having it, and she's married to this dude or having babies with this dude, and this child has it. So again, not skipping a generation. There it is in this generation, and this generation, no skipping. If we go this way, grandfather has it present in the generation, uncle doesn't, also first cousin doesn't. So you wouldn't say that's skipping a generation. If first cousin had it, then it would have skipped this generation. But it doesn't, so no skipping. And then here we have it here, father has it, mother doesn't, but sister does. So again, in every single generation, as we travel backwards, doesn't skip a generation. For things that do not skip a generation, we say they're dominant. That's the pattern. Again, we don't think about it over here because the trait of interest, it, right? I keep saying it. It's neither here nor there, so we can't use that information. It is here and it is here, no skipping. it's dominant. The rule of thumb for whether or not something is sex-linked or autosomal, and remember I said sex-linked really just means on the X chromosome. The trick here, and we haven't really talked about the sex chromosomes yet. We'll hit this heavy in a little bit, but males show sex-linked characteristics way more often than females do, and that's because they only have one X chromosome. So whatever is on that X chromosome, they're going to show. So the way that you kind of figure out whether something is sex-linked or autosomal is you look to see whether or not more males in the pedigree have it than females. Now, if you look here, it's 3 males and 2 females. That is not nearly well enough to figure out, to say, oh, it's sex-linked. That's just a rule of thumb, though. Here's another way that you can tell that it's not sex-linked, is that we've already decided it's dominant. The father has it. He's passing that on to the daughter, right? But he didn't pass it on to this daughter. If he only has one X chromosome to give, both of the sisters should have had it, but they don't. So it can't be sex-linked. It's got to be autosomal. So that's your process. We'll talk about that a lot more in lab. But you can also use pedigrees to deal with puzzles. Like, let's say you do have a breeding program and you're trying to figure out whether or not the dog or the animal that you like has a trait that you like. The trait that we're looking at here is called anury, which literally means no tail. It is a condition found in some dogs and some other animals where they're basically born without a tail. So we can see from this pedigree that, or we kind of know from other pedigrees that anury is in fact a recessive trait. So I like the way that your book kind of starts to think about whether or not this is recessive or dominant, and how you can predict whether or not offspring will have the trait versus whether they will not have the trait by looking here. So we have an individual 1 that's a female, no tail, no anury, so having a normal tail. And this one here has a normal tail. Again, it's only the colored in, the shaded in boxes or circles that mean that they actually have the trait of interest, in this case, again, anury. So neither one of these exhibit anury, that means they have a tail. Now, the only way for 2 individuals who have a trait to have an offspring that doesn't have a trait is if that trait is recessive. These 2 must have been heterozygous. So they had those 2 had 2 offspring, a male that had anury and a female that did not have anury. And so what they did was they said, well, if a dog has no tail but both parents do, like individual 3, then we know that the genotype of each one's, we know each genotype, it has to be heterozygous, right? So let's take a look and see if we can get to the next generation. This one, do we know what this one is? We know that both of its parents were heterozygous, and we know it's got a tail, but we don't know if it's homozygous dominant or heterozygous. We just don't know. So what we can do is we can mate it to something, an animal with anury. What's that called, where you take something where you're not sure about the genotype based on the phenotype, and you mate it to something that has homozygous recessive? Hopefully you remember that's called a test cross. We just talked about that in the last lecture. So basically doing a test cross right here. What are the chances of this puppy having anury? Well, if the mom was heterozygous, then it's 50-50. And if the mom is homozygous dominant, then it's 0. Just like that lesson that we learned from the test cross. So we're kind of leaving behind at this point a little bit of Mendel. We'll come back to him later, but I wanted to talk to you about some things that weren't directly related to straight up dominance and recessiveness. And so there's a couple of topics that we'll cover here. The first one is something called incomplete dominance. And when Mendel was doing all his research, it was very straightforward. You were either purple or you were white, and that was it. But with something like incomplete dominance, the heterozygote has a phenotype that's intermediate between the phenotype of the two homozygous. It literally looks like it's a shade between those two in the case of the flower colors. So incomplete dominance is what we're going to look at now. And this can be seen in snapdragons very easily. I don't know if you've ever seen snapdragons. They're really cool little flowers. And what we know about snapdragons is that color is incomplete dominant. If you cross a white snapdragon with a red snapdragon, you get pink snapdragons. That's what I was saying about the definition of incomplete dominance. The phenotype is in the middle or in between. Also, notice what your book does. It uses different names for alleles. Instead of having a big letter and a little letter, like you would with normal dominance or recessiveness, and I mentioned that if you have a big letter, that means dominant, and if you have a little letter, that means recessive. But with incomplete dominance, you don't actually have something that's dominant, and you don't actually have something that's recessive. They're incompletely dominant. And so we have to use different kinds of alleles. And the way we do this is we use this really weird little thing called a superscript. So with the superscript, that's indicating what version of the phenotype you have. And the lower letter, the main letter, is the trait that we're looking at. So we're looking at color here. So using the letter C to indicate that, that we're looking at color, and W is white. So this literally says, this allele right here, C raised to the W, is one allele. It says the color is white. The color is white. This individual is homozygous for the white alleles. This individual is red, and so we say that the color is red. So this individual is homozygous for red, so the color is red, the color is red. If you take a red individual, homozygous for red, and homozygous for white, you notice that each of the offspring are pink in phenotype, but they're also heterozygous for their genotype, a red and a white allele. Again, please understand that the superscript is very important, and it helps things be less confusing, even though it looks a little bit more confusing as you're going through this. So if you take two of these pink individuals and you cross them together, two heterozygotes, and you go through the law of segregation and you put them in their own gametes, you go back to this very interesting thing where you have a white homozygous for white, and you have a red homozygous for red, and you have two heterozygotes. Color white, color's red, color's white, color's red, heterozygotes. This is exactly the same thing that we saw before with the genotype of having 1/4, 2/4, and 1/4 for genotype. But notice that the phenotypic ratio is a little different. The phenotypic ratio much more closely mirrors the genotypic ratio, and that's because the heterozygote has their own very distinct color, in this case pink. So 1/4, this is actually a typo here from your textbook, sorry about that. It should say 1 color red to 2/4 pink to 1/4 white. Sorry about that little typo, I just saw that. Incomplete dominance is that one where the intermediate shows kind of a blend of the both alleles. There's another kind of non-dominance called codominance. And in this case, the heterozygote individuals actually literally shows both alleles. I love this picture. The father has these horizontal stripes in his shirt, the mother has these vertical stripes in her shirt, and the kid has both horizontal and vertical stripes in their shirt, so they're the heterozygote. And I know this isn't a real world example, but it's still pretty cool. And we're going to talk about codominance in humans in an actual example. But remember what I said before about how phenotype can be like an outward appearance, but it can also be anything that you measure about that individual that's a description of that individual. And what we're going to talk about is blood types. So we're going to talk about codominance and blood types. But this is where blood typing gets a little confusing, because we're not just going to talk about codominance, we're also going to talk about another concept called multiple alleles. So we're going to talk about multiple alleles and codominance at the same time. I know it's confusing. So let's start out simple. Do you know your blood type? I know mine. It's B positive. And if somebody asks you your blood type and you know it, you're going to say both this letter, either O or A or B, like me, or AB, and then you're going to say a positive and a negative. For now, I want you to forget about the positive and negative thing. That's a different gene. And I want you to focus just on the letters, A, B, or O. So for blood typing, there's actually 3 different alleles. And the way we write these alleles, again, it's a little confusing, is we use the letter I, and we notice that we have a capital I and a lowercase I. But we also have the capital I is always raised to the superscript. Hopefully you remember from when we were talking about incomplete dominance that when you don't have normal dominance or recessiveness, you use these superscripts. And that's what's going on right here. Capital I raised to the superscript A, that means you have the allele for type A. Capital I raised to a B means you have the allele for B, and lowercase I means you have the allele for O. This also talks about things that are dominant and recessiveness, because remember, capital letters are dominant and lowercase letters are recessive. So this is a very confusing system. If you can understand this, then you are really good and far ahead of the game. We have codominance between A and B, and we have normal dominance between ABs and O. O is recessive. So if you can remember that, then you're good. And that tells you over here as well. A is dominant to lowercase I, and it is codominant with B. B is dominant to lowercase I and codominant with A. And lowercase I is recessive to both A and B. Please notice that lowercase represents recessive and uppercase represents dominant, but it's the I that's uppercase and lowercase. The A's and B's are our superscript indicating whether or not you're blood type A allele or blood type B allele. So if you take these 3 and you put them in combination, you can actually get 6 different genotypes. You can get IA, or IA little I, IB, or IB little I. You can get IAIB, that codominant one that I'll talk about in a second. And you can get little I. So both of these, the ones that have IA, and I'll show you over here because it's a little bit more clear. The ones that have at least one IA, they're type A. Their blood is their phenotype. And that's what type means. It literally means phenotype, not genotype. That's what type it shows in the blood test. We'll be doing blood testing like this in lab. So homozygous and heterozygous. We wouldn't say homozygous dominant necessarily. We could, but because we got codominance going on, it's a little trickier. So homozygous and heterozygous. Same thing for B, homozygous and heterozygous. Here's where the interesting thing comes in. If you have one A allele and one B allele, your blood type is AB. You're showing both, and that's why we say codominant. If you are type O, then you have little I, you're homozygous and recessive. It would be definitely appropriate in this case. So there's 4 phenotypes, A, B, AB, and O. Why does this work? I mean, why is this very, very strange system where you've got some are codominant and some are recessive and some are dominant to others? It's just very, very confusing. And so I am going to explain the biology of the situation to you. And it has to do with chemicals, these chemicals in your body that are called antigens. Antigens are, your book says, are like signposts of your body's immune system. These are recognition proteins, antigens. And then there's these other molecules in your body that are called antibodies. And antibodies are part of your immune system too, but they're kind of more like free floating in the bloodstream. Antigens are actually attached to your blood cells, and antibodies are not attached to your blood cells. So That's one big difference. Antigens attached and antibodies free-floating in the blood. Antigen attached, blood for antibodies free-floating in the blood. I don't know if that helps at all, but that's how I remember it. So if your blood type is A, that means you make the A antigen. And if your blood type is B, it means you make the B antigen. If your blood type is AB, you're making both the A and the B antigens. And if your blood type is O, and here's the weirdness, you're not making any antigens, neither A nor B. So that's how you get dominance, codominance, and recessiveness. Making A, making B, making both, and making neither. So that's how you get AB, A, B, AB, and O. What's up with these antibodies, though? This is actually another really important part of your immune system that has nothing really to do with your genotype, but does have to do with blood compatibility and whether or not you can get transfusions of certain people's blood. And so if you have type A antigen, your body makes the B antibodies. Again, A antigen makes the B antibodies. So if you're ever given type B blood, what happens is that the B antibodies bind on to the type B antigens and basically form this terrible clot, and you die from it because the blood can't flow around in your body if you've got all these antibodies glommed onto these antigens. Same thing true for type A. If you're type B, you're making the B antigens, but you make the A antibodies. So if you're type B and you're given blood type A, your antibodies glom onto those antigens, and then they just make this huge blood clot and you die. If you're type AB, and those are your antigens, it really doesn't make any sense to make antibodies. Because if you made the B antibody, you'd kill yourself. And if you made the A antibody, you'd kill yourself, right? So you don't make any antibodies. If you're type O and you don't make either antigen, guess what? You make antibodies against both A and B. And so if you're type O, you can't get any of these other blood types because they have these antigens and you are making those antibodies. Let's take a quick look at this blood compatibility in a chart. This is a nice chart. It's good to think about, but it's also really good to kind of just use the logic that I taught you here. So if you're type A, you can make the B antibodies, sorry, and you can give blood to people who are type A, and you can give blood to people who are type AB, because they don't have the A antibodies to fight against you. But if you're type A, you can get from type A, and you can also get from type O. Notice that you cannot give to type O. Notice that you can't give to type B. Same thing here. You can't get from type AB, and you can't get from type B. Anything with a B, your antibodies, your B antibodies, would fight against and kill you. Same logic here for type B. You can give to type B and AB. You can always give to your own type. You can get from your own type, and you can always get from O, because O doesn't have any antigens on there, and so your body's not going to react against it. If you're type AB, you really can only get from, you can only give to AB, but you can get from anybody, because you're not making any antibodies. You can get from any other blood type. And if you're type O, you can give to any other blood type. Cool for everybody else, but you can only get from your own type. So that kind of sucks for you. We say that type O is the universal donor and that type AB is the universal recipient. Remember before how I was saying that if you tell people your blood type, you usually tell them two pieces of information. For me, I say I'm B positive, and that positive is a separate marker on the bloodstream called the RH marker. And this is the good thing about RH is that it's not multiple allelism. It's not codominance. It's just really, really straight up normal dominance with one gene and two alleles. Very much easier to think about than the ABO blood group. But because we talk about blood type together with the ABOs and the RH, it does get a little confusing, and I understand that. But other than that, other than the confusing part that we're going to get to, and we're going to spend some time on that in lab and in lecture, here's what I want you to know. Just like the ABO blood group, you're going to have antibodies and antigens. If you have the RH antigen, you don't make the RH antibody. If you lack the RH antigen, you make the RH antibody. So if you make the RH antigen, you're called RH positive. And if you lack the RH antigen, you're called RH negative. Again, it's a little bit of a difficult concept. I'm not going to spend too much more time on that in this in this lecture. We will delve deeper into it in lab and lecture. So stand by. Just kind of keep this in mind when we do our activities. But moving on, because we do have a little bit more to cover, the next topic that we want to talk about is something called multi-gene trait. It's also known as polygenic. Remember, poly means many, and genic is genes. So polygenic means many genes that influence a very singular trait. So if we look at the trait here that's being demonstrated, we have this idea of different heights. We have the idea of different skin colors. I also like the idea that we have different tie colors. Like those are all kind of analogies, the tie color at least, analogies for what polygenic traits are. Lots of genes going into a single trait. In this case, I'm going to focus on height. So also notice that height is got lots of genes that go into determining it. Here's this old wives' tale that says you can kind of predict the height of your child. If it's a boy, you say add 5 inches to the mother's height and average that with the father's height. If it's a girl, you subtract 5 inches from the father's height and average that with the mother's height. Like, really? That if you have siblings of the same gender, then wouldn't you be exactly the same height with that formula? So that doesn't really work. There's also this other thing that says if you take a child's height at age 2 and double it, that's how tall they're going to be. Also not really accurate. The reason that it's so hard to predict the height of somebody is because there are so many genes that go into determining height. Unlike what we were talking about with Mendel's pea plants, one gene controlled it, one gene with 2 different alleles. This one we have multiple genes. And what we know is that we've got about 180 locations, possibly genes, that influence height. For each of these, they're called loci locations. For each of these locations, they play a little role in how tall you're going to be. So if you have a lot of those loci for being taller, then you're going to be tall. If you have a medium amount, you're going to be medium. And if you have very few of them, then you're going to be shorter. For example, there's this gene on chromosome 15 that codes for an enzyme that converts testosterone into estrogen. And that enzyme influences height because estrogen helps bones fuse. And so it stops growth. So if you have more of this enzyme, then your bones are going to fuse at a younger age and you're not going to grow as tall. But if you have lots of different genes, you say that there might be this additive effect, and that's the term right there, additive effect, for all these genes kind of acting together that give you the range from very short to very tall, or anywhere in between. You have other genes they're polygenic, including eye color. We used to think eye color was just one gene, and sometimes I talk about it like it's just one gene, but we actually know now that there are at least 2 genes, probably more, for any given eye color. So that's where things get to get a little confusing. Notice that we're not doing any Punnett squares for this because it's just way too confusing to start doing Punnett squares for this. We think that autism is probably a trait that has many genes that influence it. Somewhere 10 or 20 or more, we're not exactly sure. But what we do know is that people with autism fit on a range, and that range is oops, I forgot to mention a term on this, continuously varying, like a distribution that looks like a normal shaped distribution curve. So we see that with autism too. And if you're really interested in autism, you can read a little bit more in your book about the geek theory of autism, which is how this idea that autism is genetic and that it's influenced by many genes first came into play, instead of it being, as it was inaccurately done, that people thought it was due to childhood immunizations. It's totally not. It's definitely got some genetics going on there. We just are learning more and more about that. So, so again, read your book if you're interested more in the geek theory of autism. So that was polygenic. That's where lots of genes influence one trait. But we can have the opposite too. The opposite of that is pleiotropy, where you've got one gene, but there's actually lots and lots of different effects. And the example that we're going to step through is the example of sickle cell anemia, where you have one gene with many effects. Notice that the allele names, how interesting they are, because they're showing incomplete dominance, aren't they? They've got these letters raised up to another letter that means incomplete dominance. So individuals, or codominance, it could be either one. This one happens to be codominance. So, so if you're homozygous for not having sickle cell anemia, then you produce normal blood cells, but you're also like a little susceptible to malaria. If you're heterozygous, some of your cells are going to sickle, some of them aren't going to sickle, but you're immune to malaria. You cannot get malaria. If a mosquito with malaria bites you, you will not get the parasite. If you're homozygous for, again, notice I'm not saying homozygous dominant or recessive. If you're homozygous for sickle cell anemia, then your blood cells produce the sickle shape, but you are also immune to malaria. Do you hear the fact that I'm talking about pleiotropy? There's more than one effect. In this case, the blood shape changes, and whether or not you're immune to malaria changes. So pleiotropy. Again, we're not doing a Punnett square per se, but you could. It's a simple trait with one gene with two alleles, but it's the outcome, it's the phenotype that's different, because you got different effects, you got multiple effects. The next topic that we're going to talk about is color blindness. And color blindness, as I hinted about before, is a sex-linked trait. And I said I hinted about it a little bit when we were talking about pedigrees, because with pedigrees, you can tell whether or not a trait is inherited on the X chromosome or not. So let's take a look at color blindness, which of course is inherited on the X chromosome. So the way we write alleles that are inherited on the X chromosome is we have to actually say which chromosome it's on. So we also have superscript nomenclature here. We say the big R allele is on the X chromosome, the little r allele is on the X chromosome. So you have it as X raised to the big R or X raised to the little r. Again, all that's saying is that on the X chromosome is this dominant allele, or on this X chromosome is a recessive allele. To be colorblind, you have to inherit the recessive allele from your parents. And depending on whether or not you're male or female, that looks a little different. If you're male and you get the X chromosome from your mom, you get the Y chromosome from your dad. If you're male, you only have to get one allele from your mom that's colorblind. If you're female, you have to inherit both of those alleles from both parents. And so being female who's colorblind is not impossible, but it's definitely more rare than if you're a male who's colorblind because you only have to get one version of that. To have normal vision, if you're a male, you have to have inherited that normal allele, dominant allele from your mom. But if you're female, you can kind of get it both ways. You could get the dominant allele from your mom or your dad, and same with the recessive. You could be heterozygous, in other words, or you can be homozygous. There's definitely more ways to be colorblind or non-colorblind if you're female. So males are much more likely to be colorblind because they only have the one X chromosome. And they always inherit that from their mother, right? Because they're inheriting the Y chromosome from their father. So that means that moms pass on that trait to their sons. When it comes to female offspring, it's a mix and match. You can either inherit the big R or the little r from your mom or your dad, right? Because you got to have two X's, and your dad is giving you one of those X chromosomes, and your mom's giving you the other. So that's colorblindness. But there's other traits that are sex-linked, and they're a little bit more complicated to figure out. Male pattern baldness is one of those really complicated things that it took us a long time to figure out. Remember, we're not doing any crossing our experiments, right? We're just like, you have to look at pedigrees basically. But the scientist Dorothy Osborne, she was the first one back in 1916 to actually study the heredity and cause of blindness. So baldness, specifically male pattern baldness. Back at the time, most people thought that wearing hats kind of disrupted the blood flow to the scalp, and that's what caused baldness. So she wanted to set about testing that. How would you propose to test the idea that hats cause baldness? And what Dorothy Osborne did was she went about trying to figure it out by looking at this baldness. But of course, baldness is really hard to study because as you age, you become more and more bald, right? Older people are bald, but younger people tend not to be. Although there are some that are, absolutely. And then, of course, it's possible that not all balding patterns have the same cause. So even though it was back in 1916 that this was first kind of hypothesized about, it wasn't until 2005, that's this century, almost 90 years after it was originally proposed to be studied, that we actually figured out a good way to figure out what causes or how male pattern baldness is inherited. So they took the DNA from 391 men. 201 of them were balding, and they took them from 95 families. What that means is that they were looking at brothers within a family and comparing the DNA of those brothers, especially ones that exhibited early onset balding. And then in comparison, they looked at the DNA of unrelated men, some of which were young, under 40 and having baldness, and others that were over 60 with no baldness. Think about these as the control groups, okay? So what they found, what the results were, is that men with male pattern baldness were more likely to have a specific stretch of DNA, but that stretch of DNA was on the X chromosome. And unaffected men had a different sequence on the X chromosome. And this is how they figured out, through looking specifically at DNA and not necessarily pedigrees, that this was on the X chromosome. So the data does support the hypothesis that male pattern baldness is inherited by men from their mothers. So the thing that we long suspected is true is actually true, but that it's just a region within this gene that we'd already kind of suspected. And we also know that men with male pattern baldness actually have a higher number of receptors for the male sex hormone. What this means is that they're putting out the same amount of testosterone. It's not that they have more testosterone. It's that those cells are more responsive because they have more receptors. And remember, receptors are the things that say, hey, I see you, signal. I'm going to change what I'm doing because of you. So we also know that men that are castrated as young men, for whatever reason, this was done for a long time to have people with voices to keep kind of boys as being sopranos. Men who are castrated don't actually go bald. So Game of Thrones was wrong, if you know what I'm talking about. Interesting. So the take home here is that you do see male pattern baldness in families, and that the baldness is caused by a sex-linked gene, which is on the X chromosome, and that males that inherit that allele on the X chromosome, they're definitely getting it from their mother, and so it is also true that for higher activity of that receptor gene, they're more likely to have male pattern baldness than if you have the other gene. We also know that some women can have male pattern baldness. That's if they have two X chromosomes, both with the recessive alleles. So I'm going to go ahead and stop it here, a little longer than usual. My next video is going to be much shorter than usual. Sorry about the timing difference, but it is time to stop this. So I will pick this up with environmental effects and some other good stuff when I come back.